So we're very pleased to have today with us in our Bar-Ilan University Vision Science Seminar, Dr. Rimona Weil. Dr. Rimona Weil graduated from Downing College, Cambridge, and went on to study medicine at University College London. Her PhD research at the Wellcome Trust Center for Neuroimaging, UCL, examined the integration of visual signals in healthy and damaged brain. Following this, she trained as a clinical neurologist at the Royal Free Hospital, a national hospital for neurology and neurosurgery, Queen Square. She was awarded a postdoctoral UCL Excellence Fellowship in 2014 to study the visual changes in Parkinson's disease. Today, she divides her time between scientific and medical careers. She works as a Wellcome clinician scientist and consultant neurologist at the Dementia Research Centre, University College London, and the National Hospital, Queen Square. Her clinical work is focused on cognitive disorders related to Parkinson's disease. Her research aims to shed light on how dementia happens in Parkinson's disease, using visual perception measures as early clues of cognitive involvement in Parkinson's disease. Her ultimate aim is to use these measures to inform clinical trials to slow the progression of dementia in PD. She published dozens of research papers, including in top leading journals as Science, Brain, Movement Disorders, PNAS, the Journal of Neurology, Neurosurgery, the Journal of Neurology, Neurosurgery, and Psychiatry, and others. In her research, she combines multiple methods, including advanced and more classical neuroimaging techniques, behavioral and bedside measurements, including at different levels of perceptual and cognitive processing, clinical measurements, retinal measurements, and more. Today, we will hear about her work and her research, focusing on how she uses visual clues to pick up on the earliest signs of dementia in people with Parkinson's disease. So without further ado, welcome Rimona to our seminar. We're so pleased to have you with us and hear about your research.
Thank you so much. That was the most tremendous introduction, and you've done a lot of research on that. I didn't give you that. So
I know.
That's quite an embarrassing introduction, so thank you. It's really lovely to kind of be with you, but not with you while I'm in London in the rain. Yeah, as we've sort of said, we're sort of meeting everybody who's here, people that I've heard of for like quite a long time. So what I'm going to show you today is some work that I've been doing where we've been using vision to understand dementia and Parkinson's disease. And I'm going to show you, well, in fact, I'll just show you this. I'm going to take you through sort of 4 key areas. And it's a mix of published data, but there's some fresh stuff there, and sort of just a heads up, there's some negative findings as well. So, and I'd be really interested to know what you think about this and your thoughts about all this, especially. It's really exciting to speak to vision scientists about this work, because I present work to different groups, but actually people who really get the vision side. So I'm going to start with kind of visual changes and even like why vision in Parkinson's. I'll show a little bit of retinal data and then some white matter changes, a little bit of sort of graph theory and thinking about connection types, and then some advanced neuroimaging where we've been looking at brain iron in Parkinson's. And the whole thing, it would take about 35-40 minutes. So is that about that right? Great. So, well, actually, the story of Parkinson starts in London. So it's around 200 years ago, and James Parkinson was an apothecary working in London, and he made his observations actually in 6 different patients who he noticed, 4 of whom he never met. He just noticed them walking down the street, and he made his this description that it still stands today. That it's amazing how it's held up over more than 200 years. And he described an involuntary tremulous motion and a propensity to bend the trunk forward. But very interestingly, he thought, and this is probably because he didn't actually meet some of the people, he thought the senses and the intellects were uninjured. And actually it was sort of nearly 60 years later that Jean-Martin Charcot noticed that actually at a given point, the mind becomes cloudy and memory is lost. And actually that's the Parkinson's disease dementia. And when I see patients in my clinic, that's what terrifies them. They'll say to me, look, I can control the movement disorders, the tablets are quite good, but I don't want to get dementia. So a lot of the motivation for this work is to work out who's going to get dementia, because if we could then target those people, then maybe we could at some point start some intervention to prevent it. But let's just think sort of pathologically. When we think about Parkinson's disease, we think it's classically, we think about the midbrain and sort of breakdown of dopaminergic neurons. And here I'm showing you on the left a slice through the midbrain, and the left a control, and you can see the substantia nigra. And on the right, a patient with Parkinson's, where there's that loss of substantia of the blackness because of the degeneration of those neurons. But Parkinson's is much more than a disorder of the midbrain, and it's much more than just dopamine. So actually, it's a disease that affects the whole brain, and actually a bit beyond as well, but I won't go into beyond the brain today, and certainly affects the cortex. And we know this now from several sort of neuropathological studies, and there may be a progression from the midbrain up the basal ganglia and through the cortex, although even that may not be quite correct. But certainly it's a disorder that affects the cortex, and it's characterized by these, especially by Lewy-related pathology, which will come up a little bit later. So alpha-synuclein inclusions in the cells. So why vision and Parkinson's disease? Well, it turns out that if you have Parkinson's disease and you have visual problems, you're at higher risk of developing dementia. And there's now actually convergent evidence across different countries where this has been looked at and using different techniques. So Julius Anang, working in Canada, showed that if he took people with Parkinson's and he asked them to organize these sort of colored discs or squares, the patients who struggled getting the order correct, when he then followed them up, they were more likely to have dementia after follow-up. Caroline Williams-Gray was working in Cambridge, and she looked at patients who had been newly diagnosed by their GPs, and she gave them a whole battery of tests. And one of them was, she asked them to copy intersecting pentagons. And she found that people who at baseline made errors copying intersecting pentagons, when she followed them up to 10 years later, those patients were at double the risk of getting dementia. In Michigan, Nico Bonin was using PET imaging and looking at glucose hypermetabolism. And he found that patients who at baseline had hypermetabolism, occipital lobe, visual processing parts of the brain, when he then followed them up 6 years later, those were the ones who had dementia. So early occipital hypermetabolism seems to be a risk for dementia. And then neuropathology studies. So this is a post-mortem study that was done in the States. And I want you to focus on the red line. So it's a survival analysis. And the red dotted line are patients who had Lewy-related pathology, but especially in the occipital cortex. And those patients developed a much more rapid dementia, and they died quicker. So it seems to be that having visual changes is a marker that you're going to get dementia. And actually, over now, I think it was 2020 now, this twin pair of studies was published in Movement Disorders, looking at huge population-wide studies that have really supported this. The first was in America, and the second one was in South Korea. And they took insurance data of just people who had Parkinson's disease, and then they looked at those patients who had Parkinson's who also had visual problems of any sort. So it could have been cataracts or anything. And they found that the patients with Parkinson's and visual problems were at higher risk for all sorts of bad outcomes. So that was depression, falls, breaking a leg, dementia, and death. So vision and Parkinson's seems to be bad news. But how did I get into this? Well, when I got into this was about 5 years ago, 7 years ago now. I didn't know any of this, and a lot of this wasn't published. But actually, I got into this because of a clinical observation that was made at the time to a colleague of mine, which was that people with Parkinson's disease had trouble reading capture images. So they've changed over the years. They're the things that when you go to the banking websites, they want to make sure that you're human. But at the time, there were these distorted letters and numbers, and you had to say what letters and numbers were that you saw on the screen. And this colleague knew that I had worked with Sharon and with Geraint Rees and said, oh, you've done some vision science. Why are my Parkinson's patients struggling with capture images? And I thought that was quite intriguing at the time. I thought, well, Parkinson's, it's a movement disorder, it's the basal ganglia. Why are they struggling with this? So we decided to test this. But instead of using letters and numbers, which are quite confounded by edges and lines, we took images of cats and dogs, because everybody knows what a cat or a dog looks like, but actually at the sort of image processing level, it's quite a complex task that's being done. And we did, so we actually controlled the sort of baseline levels of these images of cats and dogs by using a Fourier transform. So they all had the same image frequency, but different images. And we had a very big bank of pictures of cats and dogs. And what we did with each of these images was that we then distorted them. We did an affine distortion by varying amounts. And we showed them very briefly, and we asked patients, we gave them a 2 alternative force choice. Are you seeing a cat or a dog? And we gave them 3.5 seconds to respond. So we did this many times, so 400 trials, with a different cattle dog in every trial. And we had this image bank of 500 images I found on a sort of online resource. And we varied the amount of skew on each trial. So we had 11 levels of skew, and we randomized the order that we presented patients, the pictures. So by varying the amount of skew, we could then, that allowed us to build a picture of their performance at each level of skew. So this gives us a sort of standard psychophysical curve. So on the x-axis, we've got how much skew there is. So 0 is no skew, and they're at 100%, as you'd expect. And 5 is extremely skewed. And we get this nice curve. And what we're interested in is the halfway point, when it's halfway between extremely skewed and not skewed at all. What's the, like, how much skew can they tolerate at that 75% point? And then for a Parkinson's patient, that sigmoid curve is shifted to the left. And they're just as good, allowing for, you know, a bit of variation, but they're just as good at when the image is not skewed, but they couldn't tolerate quite as much skew, so that the threshold is pushed over to the left. So it's these thresholds that we can then compare. So when we did that with we had a small number of controls, age-matched controls, and people with Parkinson's, none of whom had dementia. We found that overall, the threshold, the amount of skew that people with Parkinson's could tolerate, was lower than what the controls could tolerate. So we quite like that, because the people with Parkinson's, they haven't got dementia. On the surface of it, they have high functioning. But what we thought was much more interesting than the difference between Parkinson's and controls was the fact that there was this wide variation. So there's some patients are doing a lot worse than controls, but there's even one person who's doing quite a lot better. So we said, well, they all look about the same in terms of their, not just what they look like, but in terms of their Parkinson's, similar disease duration. They don't have a sort of frank ophthalmic problems. So what's separating these people out, and what would happen if we follow them over time? So we did that. We looked at them over a year, and here I'm showing you the skew this time as a D prime instead of as a threshold, but it's essentially the same. So for those, and then on the y-axis, I'm showing you change in MoCA. So MoCA is a kind of global cognitive score, and what you can see is that the patients who at baseline had a lower D prime and were less good at detecting these cats and dogs that were skewed were the ones who were more likely to drop their cognitive score after a year. So we thought it's kind of quite interesting, but the numbers are small, quite like the cats and dogs test, but actually does this generalize to other forms of other visual tests? So we set up this vision in Parkinson's study, and we invited a larger number of patients to come to UCL and have a whole battery of different tests. So we did cats and dogs tests, but we also did several other visual tests. So we did logMAR acuity, contrast sensitivity, some biological motion, because I'd seen that from Aisha Sagan in the lab. So a whole bunch of visual tests, detailed neuropsychology for sort of 45 minutes to an hour across 5 different domains. We did some retinal imaging, we've collected plasma and genetics, and then we did different MRI sequences. And we did this for 100 patients with Parkinson's and 40 age-matched controls. And they attended, so baseline was in around 2017, took around a year to get everybody through. 18 months later, we did visit 2, which was we had 10 short, and then COVID hit. And then we restarted in April 2021, and we're nearly, we're temporarily paused, but we've got another 15 or so patients. So we're nearly finished visit 3, so sort of out to 3 years. So I'm going to start by showing you some of our baseline data. And at the baseline, of course, we didn't at that time have any follow-up. So if we want to think about, well, how do these visual scores measure up? We don't know what's going to happen to them cognitively, but what we could do is, at the time, there had just been some risk scores for dementia and Parkinson's published that had just come out. So one of these used a bunch of clinical measures, so age at onset, because actually the older you are at diagnosis, the higher risk of dementia. And then some other clinical measures, so MMSE is a cognitive clinical score, years of education, UPDRS3 is a movement score for Parkinson's, depression. I've put GBA status in brackets, that's a genetic test, and at that time we didn't have any genetic data. So that was one, this was a kind of algorithm where we could combine these measures to get a risk of dementia. But there were a couple of other dementia risk scores that we actually replicated our findings with. One of them was from Caroline Williams-Gray. So she just used 3 measures, age at onset, a sort of shortened version of this motor UPDRS score, and how many animals you can name in a minute, which amazingly is actually a pretty good risk score with just those measures. And Annette Schrag, in actually the same issue of the Lancet, had another algorithm with similar measures, but some of them were a bit different. So we had these risk scores and we had our vision scores. So how did they measure up? Well, we were pleased to see that actually the cats and dogs test did look like it was holding. So here I've got on the x-axis performance in cats and dogs. The lower your score is, the less tolerance you have for skew, so the worse you're performing. So the lower score, the higher risk of dementia at baseline. But we've done things like acuity, actually mainly to check that patients weren't frankly blind. So we would have excluded sort of very, very poor acuity. But to our surprise, actually visual acuity also correlated with risk of dementia. So this is a logMAR, so the higher score of acuity actually means you've got poor acuity. So again, poor acuity correlated with risk of dementia. But we'd also collected this retinal imaging. So for people who know a lot about retinal imaging, apologies, I'm just going to explain it in case there are people who don't. So this is me having optical coherence tomography done on me by Louise Leyland, who is my postdoc. And in the background of the picture, you can actually see that's my retina. And you might be able to tell I'm smiling, so it doesn't hurt. It's a painless experience, and it essentially works by bouncing light waves through the pupil off the back of the retina. And apologies for people who know a lot more about retinal imaging than that, but that's sort of the gestalt. And what that does is it gives you a 2D buildup of the retina, and you can get the volumes of each of the layers. So I'm going to highlight three particular layers. So the slightly deeper ones are the GCL, ganglion cell layer, and the IPL, the inner plexiform layer. And we were particularly interested in those two layers in the context of Parkinson's for two reasons. So one of the reasons is that within those cells, there are amacrine cells within those layers, there are amacrine cells that kind of spread, extend out, that actually use dopamine as a neurotransmitter. So we thought sort of slightly simplistically, well, Parkinson's disease, dopamine in the retina, maybe that will be implicated. But it's even stronger evidence for using those two layers is that at post-mortem, that there are post-mortem studies that have looked at retinal layers and the brain and showed that Lewy-related pathology, so alpha-synuclein, specifically phosphorylated alpha-synuclein, builds up in those two layers, in the GCL and the IPL, rather than in the other layers. And that also the amount of phosphorylated alpha-synuclein correlates with the amount of alpha-synuclein in the brain. So it seemed like, well, then maybe those layers are affected in Parkinson's. The most superficial layer is the retinal nerve fiber layer, and we thought that's least likely to be affected. So then, I'm going to show you, I'm showing you here that for the IPL, just because to fit it on the slide, but it's exactly the same for the GCLs. I'm not hiding anything, but it's exactly the same. In fact, I've since learned that you can combine IPL and GCL layers. So what we found was that patients with thinner inner plexiform layers showed a higher risk of developing dementia, but we didn't see that relationship in the RNFL, in the retinal nerve fiber layer. So, and this is what we were talking about at the beginning with Professor Mandel, that people have been quite excited that, could the retina be maybe a biomarker for dementia in Parkinson's disease? It's very easy to, lots of people have got OCT machines. It's very easy to measure them. You can pull the numbers out quite easily. So the big question is, well, does this hold up over time? So around the time that we were starting to analyze our data, a Spanish group published a really nice paper in Annals of Neurology showing that retinal thickness so they combined the GCL and the IPL into one layer, which is actually quite standard to do. And they showed that patients with thinner GC-IPL at baseline had a lower MoCA score when they followed them up. And this is at 3 years. But they even showed that the ones with thin in the sort of lower tertile, they even showed actually at the 18-month that there was a little bit of difference between patients with a thin GC-IPL than the ones with the thickest. So it's a very nice paper, and they really showed very nicely that baseline retinal thickness does relate to cognitive scores, and they even showed it related to motor scores. So we got excited and thought, well, let's look at our data. Now, so far we've only got 18-month data. We're nearly done, so we'll see what happens at 3 years. But actually, we didn't find this at all. We found no relationship between baseline GC-IPL thickness and follow-up cognitive scores or movement scores, and then we even looked at more detailed cognitive scores that are a little bit more sensitive. We didn't find any relationship at all. I'd be interested to know what people think, maybe when we talk after. Is this because it's only at 18 months? Maybe when we look at our 3-year data, we'll start to see a stronger relationship. Or maybe it's just not there. So I'm starting to wonder, maybe if we want to look at biomarkers and if we want to sort of have measures of progression of Parkinson's disease, maybe it's not strongly, robustly in the retina. We probably need to go back to the brain. So brain imaging and Parkinson's disease. Parkinson's disease is really common and loads of people have got MRI scanners. So there are many, many imaging studies of looking at risk of dementia and Parkinson's disease, but they show very inconsistent findings. So looking at Parkinson's dementia itself, all sorts of regions have been implicated, and actually it's very inconsistent. So studies showing the insular, hippocampus, frontoparietal regions, and it's even more inconsistent at early stages at MCI, or mild cognitive impairment, and really, so different regions, parieto-occipital, medial temporal. Joanna Pereira's shown actually regions across the whole brain, and also precuneus. And actually, this is something that we reviewed recently. So we're just not, so we're looking at straightforward MRI sort of volume shows inconsistent findings when you're trying to relate that to dementia and Parkinson's disease. But maybe some of that is because we're looking in the wrong place. And actually volume that's measured on MRI is when you see atrophy on an MRI scan, that most likely represents neuronal death. And actually widespread neuronal death is, if anything, it's a very late event in Parkinson's disease. So if we want to monitor Parkinson's disease, looking at measures that track neuronal death are just not going to be sensitive, especially not at early stages. And from animal models, it looks like actually it's much more likely that it's axonal changes where we see differences much earlier on. So, and this is data from mice and from rats, that actually the earliest changes are axonal pathology, and you see them well before we find neurite loss. So if we want to look at axon changes in humans and in vivo, the best way to do that is to use diffusion imaging. And the conventional way to do diffusion imaging is diffusion tensor imaging. And that, I'm showing you here that, I mean, that's pretty good. It works essentially on movement of water molecules. And when the neuronal fibers are, I have to talk with my hands, when the neuronal fibers are lined up, the diffusion tensor is pretty good and it can estimate the principal direction quite well. The problem is that when the fibers are crossing, it really falls down, and you can see it cannot estimate the principal direction. So I'm going to show you some work here by Angelica Zarkali, who was my PhD student, and she's now hanging around as a postdoc, which I think is the best compliment ever. And you'll see she's done fantastic work. So she used a higher order model to look at diffusion imaging. She used something called pixel-based analysis. There are other higher order models as well, but this is one that she used. And to do pixel-based analysis, she estimated the fiber orientation distribution function. And when she did that, you can see that for the parallel fibers, the estimate is just as good. But where it really does well is it gives a much better estimate of the crossing fibers. And 90% of the brain are fibers crossing. So if you want to measure white matter changes, really we need a technique that's going to get the crossing fibers. And so what you can see when we do that, we get for each, I mean, the pixels are considered like a sort of equivalent of a voxel. That's why they use the term pixel. So we get really nice estimates of the direction of the nerve fibers, well, not the nerve fibers, but the direction of the bundles across the brain. And then just to sort of, when you think about how a nerve fiber can change, it can become less dense. So we can look at fiber density. It can become, the cross-section can become thinner, fiber cross-section, and you can get a combination of fiber density and fiber cross-section that can both become affected. So that's the FDC. But actually, I'm going to mostly show you data from fiber cross-section. We found it affects across all three, but actually that was the most sensitive for our patients. So Angelica looked at Parkinson's patients compared with controls and just looked at, well, what were the white matter changes across both of them? And what you can see is there wasn't very much. So a few ditzels kind of near the midbrain, but really not very much across comparing PD against controls. But we had all these visual measures. So I mentioned that we've got cats and dogs task, but we've also got biological motion, and that probably most of you will be very familiar with. So it's essentially dots at the major joints of a person, and it gives a very strong percept of a person moving. So here's somebody jogging, and we can adjust it psychophysically by adding more and more dots. So we can work out the threshold of where they can only just tell that it's a person moving, rather than just random dots with the movement sort of controlled against it. So we showed patients either random dots moving or a person moving, and we asked them, what are you seeing? Is it scrambled or a person? So because we weren't completely hung up on cats and dogs, and we really wanted to capture the patients who would absolutely had greatest difficulty with higher order vision, we took the median performance for both the cats and dogs and the biological motion task. So anybody who was below the median, the group median of both tasks, and we called those patients the low visual performers. And when we compared the sort of demographics of these two groups, they're pretty well matched. I mean, we captured, so 34 patients were in the low vision group, 70 are in the high group, but they're matched for age, their MoCA, so that's that cognitive score, the UPDRS, their movement score, disease duration is the same, and LEDD is their levodopa dose. So that's about the same. There were more men in the low vision group, and actually that didn't surprise me at all, because actually there's quite a lot of evidence now that men with Parkinson's do tend to do worse and are higher risk of dementia. So there were more men in this low vision group, and we then controlled for, well, we actually controlled for age as well as gender in our analyses. But they're pretty well matched. But then when we then compared our low vision patients with our high vision, so the high vision of people who were good at the visual tasks, where before we had barely seen any differences, we started to see some very significant differences in white matter between these two groups. And because we were interested in visual changes and how those might be affected, Angelica then also looked at 11 tracts that were related, that were linked to visual areas. And she compared these high visual performers and the low visual performers and looked at the fiber cross section between the percentage differences between these two groups for these 11 tracts. And what she found was that several of these were reduced in the low vision group, particularly tracts connected to the thalamus, so the posterior thalamic radiation as one example. So that's at baseline, but we do now have some longitudinal data. So what does happen to these patients when we follow them up over time? So I'm just going to move the Zoom thing. So what we found was it did hold up. So people who were poorer at the visual tasks, and remember they're defined by worse performance on the cats and dogs and the biological motion, they were more likely to develop PD-MCI, which is Parkinson's with mild cognitive impairment. They were more likely to drop their mini mental state examination score, so that's a cognitive score. And baseline performance on the visual scores correlated with this kind of more detailed cognitive score at follow-up. So it did hold up. And actually, you do all this work, you don't know, is it going to work? But actually, unlike the retinal data, visual performance, especially higher order visual performance, was really holding up. That if you're bad at these visual tasks at baseline, those did seem to be the people who were dropping cognition. And what about in the brain? Well, I'd already shown you that at baseline, the poor visual performance had these white matter changes. When we looked after follow-up, there was really much more, even more widespread white matter changes across the whole brain in these patients who were poor visual performers compared to the ones with retained sort of high visual performance. So we've looked at baseline data, we've looked at poor vision predicting dementia and Parkinson's. So the next question we had was, we're quite interested in these sort of white matter changes. One of the things we wanted to know was, well, what is it? Why are some fibers more affected? Is there something that, what is the reason for selective vulnerability of particular parts of the brain to neurodegeneration in Parkinson's? So, oh, sorry, I was setting up the next question, but just to say that for all the visual tracts that we looked at before, all of them were reduced between the low visual performance compared to the high visual performance. Right, so selective vulnerability, why are some tracts more affected than others? So to answer this question, we did some graph theory analysis, and I was going to explain graph theory, but if this is, I hope this is not too straightforward for people, but essentially graph theory is a way that you can look at connections. And really, I think the person who really got graph theory is Harry Beck. So he was the man who designed the map of the London Underground, and he realised that if you want to really understand the London Underground and make it really very clear to people, especially people who haven't used it before, then we don't need to know what the map looks like on the ground. It's actually much more important to know the connections between the different stations. So that essentially is what graph theory is. It's about the connections, the direct connections between hubs or stations or whatever your nodes are. And we can do this with brain imaging by taking the grey matter image and parcellating it into regions. So we used the Glasser Atlas, so that gave us 360 cortical regions and 19 subcortical regions, so 379 regions altogether. And then we've got our white matter image where Angelica has done the pixel-based analysis. So we've got a very nice tractogram and we've got all the connections. And then for each of the connections, we can then take, for each of the regions of interest, we can look at, are there connections between them? And then we can then build up a matrix of, is there a streamline? And then it's actually a weighted matrix using a multiplier by how strong that connection is, by a sort of cross-sectional multiplier. So this is the 379 by 379 regions, and each of those letters are a lobe of the brain, so frontal, temporal, occipital, down to subcortical. And what you can see is that there is structure within that structural matrix. So regions from the same side, from the same region, are particularly well connected to each other. So we get a very nice diagonal. And also the subcortical regions are well connected everywhere, which is why we get the sort of edges to the matrix. So now we've got our matrix, if we want to do some analysis on it, this is actually also work that Angelica performed brilliantly. She then said, well, we've got all these different nodes, let's group them into the closest communities. And the way that she did that was using a Louvain algorithm, which essentially says, let's group the communities that are most connected to each other. And what you can then do is build up a sort of number of the communities that you have. So you can choose different numbers, and the number that she chose of the number of communities here, I'm showing you 9 communities, but she's also replicated this with different numbers of communities. So you can have, we've replicated this with slightly lower, with 6, and also with 10. And for each of these communities, we then say, okay, what are the different types of connections? So we're going to be looking at connection type. Are particular connection types more affected in Parkinson's who have visual problems? So there are 4 kind of connection types that you can think of. There's interhemispheric, so going across the hemispheres. There's intrahemispheric, within the hemisphere, going from the subcortex to the cortex, and then intramodular, so within the module itself. And her question was, is there a difference in the connection strength, essentially, between these different types of connections? And what she found was that there was quite a strong difference. So for the interhemispheric, across the brain, and subcortical cortical, she found several significantly reduced connections that were significantly reduced in connection strength. But she found the opposite. So for intrahemispheric, either there were no reduced connection strengths, or if anything, a few of them were even slightly increased, and especially for the intramodular. So particular connection types, so interhemispheric and subcortical cortical, seem to be more affected. So that's the type, but what about length of connections? So are shorter or longer connections more vulnerable? So to look at this, you looked at streamline length, and it's quite clear that actually certain connection types are likely to be longer than others. So subcortical cortical are very long, and intramodular are the shortest connections. But actually, even within interhemispheric connections, some are longer than others. But within each connection type, there was a weak but still significant relationship where the longer the connection, the more connection atrophy there was. So longer connections seem to be more vulnerable. And then when she took it back to our vision scores, she found that particularly just for the interhemispheric and the subcortical, the longer the connection, the poorer there was a relationship between visual performance and longer connections in patients with Parkinson's disease. So it seems to be that those patients who had poorer visual performance also had greater atrophy related to connection, for the connection length. So what we've looked at so far is vision seems to predict dementia and Parkinson's, that it particularly affects, it's rather than volume change and neuronal loss, it's the axons that are affected, and longer connections seem to be affected, and also particular types. But for the final part of the talk, I want to think a bit more about tissue changes. And there's a new form of, if you want to look at volume change, if we just look at volume, it's a very crude way of looking at changes in the brain, as I've mentioned, that neuronal loss is a very late event. But there are changes that happen within the tissues that would be very good if we could detect. And one of those changes is the buildup in iron. So all of us, as we're aging, are accumulating iron in our brains. And there's lots of reasons for that. Some of it is because our blood-brain barrier is leaking a bit. Some of it is to do with inefficiencies of the way that we store and we metabolize iron. But people with Parkinson's particularly accumulate iron. And they particularly accumulate iron, especially in the basal ganglia, but also in cortical regions. And and specific to Parkinson's as well, alpha-synuclein aggregation seems to be triggered by ferric iron. So, and part of the reason for this maybe is that iron leads to excess free radicals, and that in itself causes oxidative stress, which causes a vicious cycle, so that there's more and more damage to proteins and increase in pathological protein accumulation. So can we detect iron in the brain? Well, it turns out that we can, and we can use a method known as susceptibility. So susceptibility is any material's tendency to interact within a magnetic field. And the main sources of susceptibility are tissue and blood iron, also myelin, but it's mainly iron. And that's captured in the phase image of susceptibility weighted imaging. So this is work that another of my students did. This is George Thomas, and he did this together with Julio Acosta Cabronero. And also I'm going to show you some work that was supported and done in collaboration with Karin Shmueli, who's also an MRI physicist. And it's a technique called quantitative susceptibility mapping. And I just want to share with you some of the pain, because actually you kind of see colored blobs in the brain, but actually the way that we get to this is actually quite involved from a physics point of view. So the SWI, or susceptibility weighted image, that comes off the scanner is really ugly. So it has especially problems around the area between the sinuses, the air and tissue regions. And there's a lot of wrapping as well, which is a sort of physics term, where that needs to be removed. So there's an unwrapping phase that needs to be performed. And there are very, very strong background fields that need to be removed so that you can actually see the underlying susceptibility changes. And then actually the most involved and complex part of quantitative susceptibility mapping is working out the underlying dipole from the image that you have. And there are different ways to do this. The technique that we use for this first bit of work is something called multi-scale dipole inversion that Julio has been working with, but there are other ways that you can do it. And once you've gone through those stages, what you can start to see is actually quite a nice map that's really starting to show you structures of the brain with differences in susceptibility. And what jumps out straight away is that even in a control brain, that basal ganglia has increased susceptibility, but you also do see some bits in the cortex as well. So when George looked at this in our patients with Parkinson's disease, and I'm showing you here whole brain data, it's uncorrected, but it's whole brain, so there's no regions of interest here. What he found was that he took the motor scores with people with Parkinson's. So people with poorer motor scores had increased susceptibility within the basal ganglia, so in the right putamen. And then when he looked at cognitive performance, he found that poorer cognitive scores correlated with increased susceptibility in the thalamus and in the hippocampus. And what was so striking about this was that, first of all, this was without any regions of interest. But if you look at volumes in the same patients, you don't get any sort of localizing effects of volume change. So it seems to be that this technique of detecting iron buildup in the brain is probably a much more useful and much more realistic biomarker if we want to really measure and quantify changes in people with Parkinson's. But then George then said, well, can we use this measure to tell us something about the mechanisms that drive neurodegeneration in Parkinson's? So to do that, he made use of the Allen Atlas of brain imaging. So this is an amazing resource. It's a publicly available resource of 6 people in America who had died and given their brains up to science, who did not have any neurological or psychiatric disorders, certainly didn't have Parkinson's disease, and their ages range between about 44 to late 80s. And crucially, what they've done with these brains is they've taken 180 regions and they've extracted gene expression data from these 180 regions. And crucially, it's within MRI space. So what this gives us access to is gene expression data in health. I mean, they've died, but they were healthy, and it within MRI space. So George could then take the gene expression data. So he's got 180 regions, and it's over 15,000 genes. And he then could relate that to the QSM scores, so the regional differences in susceptibility and brain iron in our patients. So what we're looking at is, and this is what I should say, is this work was with Karin Shmueli and also Mina Wrighton, who's a geneticist. But what's really important to point out here is that what we're looking at is the regional variation in iron in patients and how that maps onto gene expression in health. So it can only tell us about selective vulnerability. What we cannot answer is what's happening to gene expression in patients, because thank goodness our patients have not died. So we don't have gene expression data from our patients. But what we can tell us about is selective vulnerability. Why is it that those regions are building up iron? So he used the partial least squares expression to find the combination of genes that best explains the distribution that we're seeing in our brain, in our patients' brains. And for our data, the second PLS weight was the best explanation. And what he then looked at was the top proportion of genes, so the top 20% of genes that best explain that relate to this distribution of brain iron. And we end up with this a list of upweighted genes that are upweighted in health, but they tend to be these kind of funny letters like DDAH1. We don't know, who knows what that gene does. So to interpret the genes, we use something called GO, or gene ontology analysis, and it's essentially it's a dictionary of underlying processes that these genes are involved in. So when George did this, and he looked at the regional variation of the brain iron and how that mapped onto gene expression data, he found that in health, the genes that were more highly expressed in those regions where we found more iron were related to two specific, overall two groupings of processes. So this is a sort of semantic map. It's a visualization called Revigo for displaying the processes. And the two groups of processes were detoxification of metal ions and synaptic signaling. So what this is suggesting is that, and in a way, it's not surprising, but we didn't even hope in our wildest dreams that we would see gene expression related to heavy metals, because usually that wouldn't be the gene processes that would come up. So suggesting that those regions where we're seeing buildup of brain iron are regions where there's greater expression in health of genes relating to detoxification from heavy metals of all sorts, of copper, and of inorganic compounds. So for the very final part of the talk, I've talked about that we've seen differences in gene expression, but differences in gene expression can also be brought about by differences in cells that happen to build up in those regions. So, for example, astrocytes will express genes differently. So astrocytes may have a particular combination of genes. A glutamatergic neuron will have a different combination of genes that it will naturally express. And the same with an oligodendrocyte, it'll have a different combination of genes again. So in any particular region, if you find a particular set of genes, that may be because there are more astrocytes or more oligodendrocytes. So the final question was, are we seeing, can we build up a map of the different cell types that tend to be expressed in these different regions based on the gene expression that we're seeing? And this is an analysis called expression-weighted cell type analysis. And when George looked at this, he found that there were 4 particular cell types that were more expressed in health in the regions where we're finding more iron. And the 4 particular cell types were astrocytes, GABAergic and glutamatergic neurons, and oligodendrocyte precursor cells. So together, he's used this advanced imaging technique, quantitative susceptibility mapping, to then start to unpick and understand really the processes that are driving neurodegeneration in our patients with Parkinson's disease. So just to bring it all together, I've shown you sort of 4 overall kind of themes. I've shown you the work that we've done using visual changes and relating that to the risk of dementia. We looked at white matter changes in the brain and at baseline and then also over follow-up. We've looked at connection types and connection length, and actually that those being important factors as making particular connections more vulnerable. And then we've looked at how brain iron can track disease severity, but also might shed light onto really the underlying causes of neurodegeneration in Parkinson's. So particular thanks to Angelica Zarkali and George Thomas, who've done nearly all the work that I've shown here, and also the rest of my team, so Louise, Evelyn and Rohan, and fantastic collaborators in the UK and also around elsewhere. And thank you so much for listening, and look forward to hearing what you think.
Thank you very much, Rimona. Wonderful talk, and I want to invite everybody to unmute yourselves, and let's give Rimona a big applause for this exciting talk. And I've got my own questions, but I'm opening the floor for questions, so feel free to unmute yourself and ask.
Should I stop share? I think I'll stop share. So
yeah, you could.
Yeah, brilliant.
Okay, so I'll start. I see that there are I'll start with my first question, although I've got a few more. You showed in the beginning, you showed that the visual acuity was linked or in correlation to PD severity, but you didn't show how predictive it was later on, if I recall correctly. So was it
right,
not?
So actually, So we didn't even look at it. We didn't put it in the paper. And then when I was talking about thinking about this talk, I thought, you're going to ask me about acuity and contrast sensitivity and what happened over time. And I realized we hadn't looked at it. So we went back and looked. And so for contrast sensitivity was predictive for MOCA and the more detailed cognitive score. Acuity, which is, it's a little bit noisier, was as well for the MOCA, for one but not the other, but essentially both of them were. So it wasn't just the higher order scores, but actually also even acuity, but certainly contrast sensitivity. And actually that fits with the population work, actually, that visual changes, you know, there's not a particular test, which I went into this thinking maybe there'll be one test, but it isn't. It seems to be that visual changes overall are predictive of poor sort of cognitive outcomes in Parkinson's.
Okay, wow, interesting. Yeah. Okay.
And I wish I put it in the paper.
Well, you can have another one.
Yeah.
Yossi, do you want to have?
Yes, great talk. Thank you very much.
Thank you.
I would like to ask something about the relation between visual acuity or visual performance and dementia, and maybe there is a kind of a causative effect, because we know that people with declined vision do get worse, even with a cataract, yes? They do perform later on. There is a kind, maybe because of loss of input or things like that. So my first question, did you try to see that there is no ocular things? And what do you think about the cause and effect paradigm?
Yeah, so really important question. And so what I didn't say is that every single person who came through at baseline and also at 18 months, and now we haven't done it at COVID, but baseline and at 18 months, they all were seen by an ophthalmologist. So none of them had severe cataracts, so we would have excluded them. So anyone with severe ophthalmic disease was excluded from the cohort. And I think that's a really important point. The other side is that you've mentioned that cataracts and poor outcomes. I've actually just recently been looking at hallucinations and cataracts, and actually people don't think about treating the cataracts can improve outcomes. So there's actually a kind of really nice side of the story, that a cataract is a relatively minor operation compared to some of the quite strong medications that you sometimes need for hallucinations, sort of antipsychotic treatments. So yes, there's a treatment implication. And then cause and effect. So I think it's a complex. I think a big, because there's brain and eye. So I think hopefully I've shown you that with the white matter changes, we're particularly seeing connections between visual areas. So I think that there are brain changes that are causing some of the higher order visual problems, but there are certainly ophthalmic causes as well. I think I'm less persuaded about the retina. There may be some retinal changes, but it's perhaps a bit noisy. I don't think cataracts are caused by Parkinson's, as far as I'm aware. They're not caused by it. But you're right, if you are going into dementia and then you also have cataracts, then that could also be adding to your cognitive change. You're going to have less, sort of more noisy sensory inputs. And then, of course, that starts to go into the sort of territory of hallucinations as well. So I think visual changes do contribute to cognitive change. Where it's most important is in the brain, but it may also have some impact from the eye as well.
Mona, I have another question. I mean, I'm sure, I'm just gonna ask it. So I wanna ask how specific you think this is to Parkinson's disease. I mean, let's say Parkinson's disease and dementia are correlated, but it could also be that dementia is correlated with other conditions. I don't know exactly. I mean, I'm not an expert, but I'm thinking whether this visual or the vision related indications, are they related to dementia in general, or are they specific to the relationship between PD and dementia?
That's a really, really good question. I also didn't mention that how common dementia is in Parkinson's. So it's 50% at 10 years. So it's not like a sort of unusual thing. It's common, but it's more common in certain groups, and it's more common if you've got visual changes. So the short answer is I don't know in Alzheimer's. I think it's less related. So these kind of very kind of large scale population studies have been really about Parkinson's. So I think it's there's less of a close relationship. But Yossi's point about if you've got dementia, let's say if you've got Alzheimer's and you've got cataracts, it's going to worsen your outcomes. I think there is an effect as well, but I think that the sort of brain changes are more closely related to Parkinson's. And the reason I think it's a bit more specific is that there is this form of Parkinson's this disease that's related to Parkinson's but different, called dementia with Lewy bodies, and that it's within the spectrum of Parkinson's disease, but the type of dementia that you get, you particularly have visual processing problems. So in a way, I think Parkinson's is like a form fruste of dementia with Lewy bodies, where there are visual processing problems, and that's why I think it's more closely related.
Okay, thank you. There are questions in the chat, so if there are people now here in the audience who want to ask before I start reading them.
I'd be happy to.
Adam, yeah, sure.
First of all, great talk. Thank you very much. I was wondering if you tested for and found any relations with the with hallucinations in Parkinson's disease.
Yeah, so I love hallucinations. I'm very, very interested in them. We ran a whole bunch of questionnaires as well. So we used a particular questionnaire that measures severity and frequency, and sort of an intensity. And actually, in parallel to this work, we've been doing a whole lot of work about hallucinations, and a subgroup of the patients with visual changes also had hallucinations, but it's not a perfect match. So there were some who were not low visual performers who hallucinated, and there were some low visual performers who didn't have hallucinations. But yes, so we have looked at hallucinators, and yeah, there's so we've done some very interesting work looking at sort of mechanisms of what's driving those hallucinations. The short answer is that the white matter changes that we're seeing are that I showed you in the low visual performers. We also found similar white matter changes in the hallucinators, and then more recently we've been looking at changes in the thalamus as well. So there seem to be particularly thalamic changes in the hallucinators, and then we're very interested in sort of top down versus bottom up processing in the hallucinators. And one of the things that we've been seeing is sort of behaviorally over reliance on behavioral priors in that group. So yeah, they're a fascinating group of patients.
Can I ask a question with regards to eye tracking? When you were you aware of fixation? I mean, obviously, there's lots of stuff in the literature about pursuits, decades, gain, divergences. Was there some sort of control on that when you were doing the visual processing thing?
Yeah, no, that's a really good point, Avi. So you do get eye movement changes in Parkinson's. What they tend to get is hypometric saccades. So it's not like the kind of gross changes that you find in progressive supranuclear palsy, but the hypometric saccades. And we looked at doing eye tracking, and then we in the end we decided against it, because and ideally, like in a control group, that's what you would do, because you want to absolutely make sure that people are fixating. But we felt that in the patients that it was going to be it would be more discomfort, and it would actually just make that we'd get worse, sort of noisier results. But the cats and dogs task is it's a brief presentation. So the idea there being is that they have to fixate centrally, because that's where they're going to see it.
Yeah, unless they've drifted off, and then you're projecting into the periphery and not into the center. True. And then anything, all the different things that aren't foveal would show as poor performing.
Yep, I think that's a very good point. I think, could it all be because they're not fixating? It's possible.
Sorry, I wasn't.
No. I think it's a good point. We should probably do the eye tracked version. Yeah, we didn't specifically control.
Thank you. Thanks for a wonderful talk.
Thank you.
Rimona, Eliran Tal asks whether the distribution of ages were the same for the controls and the PDs. I think that was written in the dogs and cats task, and I think you might have even mentioned that, but
yeah. So they were age matched for all parts. So they are the same age, because age is a massive factor, and I've already told you that older people are more likely to get dementia. So it's really important to control for age. Okay. And then all our imaging analyses were controlled. You then correct for age as well to be absolutely sure.
Okay, lovely. OM says fascinating, and Anya Holbert asks, I mean, you're here, but I can read it.
Yeah, I can. Yeah. Hi, Sharon. Thanks, and thanks, Rimona. Brilliant talk. Wonderful. No, I just wondered if in your data, if you found a correlation between the retinal anatomy, the thicknesses on OCT, and any of the anatomical or functional changes in MRI. So, is there evidence in your wonderful corpus of data for that relationship between what's going on in the eye and what's going on in the brain, apart from all the vision measurements you did?
It's a great question. So we've looked at retinal data and volume, and there's no relationship. We haven't directly looked at correlations with diffusion, but one of the things we have done is we've done some kind of, I've been working with a computer sort of modeling person. We've looked at order of events, because that's, again, something that's quite interesting. Yeah. And if I remember rightly, it's been published. So Oxtoby is the first author. I think the retinal, because one of the things we wondered, is it brain first or retina first? And it's got to be brain first. So the retina is kind of noisy and it comes out a bit later. But directly looking at diffusion in the retina, we could look at that. I mean, I think it won't be very strong, because I think that whether it's just our data or quite what it is, but I think the retinal data are quite noisy. But it's a good point that we should look.
Okay, thanks.
Thank you.
Lovely. Okay, so Elena Adjus Vela asks, what about eye movements in Parkinson's? Are they affected by the Parkinson? If so, can they explain visual problems? Which I think you may have also already talked about or answered to Avi. Do you want to say it yourself, yeah. Elena?
Yeah, you basically already answered to this question to the other person. And also, I want to know which kind of feature, because you show the data on cats and dogs and basically acuity, but can cat and dogs be a problem to a specific area of the visual system, like the face area, or is it more, what is that really is affected? Is the global shape, the feature of the shape, what they cannot see, or what they cannot process in the visual task?
So I think that's a really good question. So yeah, what are they struggling with? I think the first thing is, I don't think it's specific to the cats and dogs, because we found also this effect with biological motion and with contrast sensitivity. But if we want to know, well, what is the brain area? I mean, I did have a bit of a look at that. We did actually put people in the scanner and look at, do functional MRI while they were doing cats and dogs task. And actually, in the end, it didn't, because I was wondering, is this a sort of like LOC, or is FFA, as you were saying? Actually, it ended up being quite sort of non-specific and possibly maybe precuneous, which, if anything, goes more with the results that we found now with the imaging, with the structural imaging, that actually it's more kind of connectivity changes in a kind of more gross and more global way than a particular brain region that's impaired in those patients. But I think that's a really good point.
Okay, and the other question, I'm not an expert in Parkinson, so I don't know how much big are the eye movement in Parkinson's disease. Can they affect a lot of the visuals? Only the eye movement can produce a problem to the visual procedure, or you think that the visual problems are unrelated or bigger than the eye movement in Parkinson's? Because people that have astigmatism, they move their eye, but they are not dementia. So I was wondering the relationship between eye movement and visual processes.
Yeah, so I mean, they do, like I said, they do have eye movement problems, but I don't think that explains all the visual changes. So I think there is a higher order visual problem that they have, and we see that. I mean, we also do kind of more kind of pen and paper type questions, tests with the patients. So things like fragmented letters, and the patients with eye movement problems can interpret fragmented letters. But if you've got higher order visual problems, then you would struggle with those. So I think it goes beyond just the eye movement. So I think it's a good point.
Yeah. Okay. Last question: do they maintain the periphery and fovea organization? So stimuli in the periphery and the fovea are differently processed, or with the Parkinson, this differentiation is affected, or?
So actually that's a very good point, and it is something that we've looked at I've looked at it, and actually there's been some work. It's quite old now. It's like a really nice one. I've forgotten the name of the guy. I think it was Lee is the author, that looked at central fixation and then a task in the periphery. And patients with Parkinson's do have particularly impaired peripheral vision. It was a very nice task that he got them to kind of, I think, dial down the peripheral how much contrast they needed in the periphery. And we've actually done this using a web version, a web-based test. And the way that we controlled eye movements there is that we did a mismatch in the sizes. So we asked patients, we showed pictures of animals that were more like line drawings, and we showed them at center fixation and in the periphery, but the central one was tiny. So they had to fixate, they had to foveate on the sort of the center, otherwise they wouldn't be able to do the task at all. And then by doing that, we've shown that there's poor peripheral vision. And actually, it's quite nice because one of interpretation of why do hallucinations happen and why do patients get these passage hallucinations where they see things in the peripheries is that they have worse peripheral vision. So that's a very good point, that there is this discrepancy between peripheral and central vision in Parkinson's.
Can I have another question after I stop myself? I read that blind people, there are no blind, recording of blind people with hallucination. So I was also wondering if there can be a relation, which is the relation with congenital blind and maybe Parkinson's disease, because if in some way blindness help hallucination, in the other way, blindness with a problem, visual problem affect dementia. So I want to know if you know, because I didn't find anything about blindness and dementia. So I want to
blindness is a fascinating question, because actually there's a completely separate condition where blind patients or very, very visually impaired patients get hallucinations called Charles Bonnet. And there was a paper, was it from here? Was it from you guys?
I think it was Avital,
yes,
Chachami, and
yes.
Rafi Malach, and maybe additional collaborators.
Yeah, so they were looking at exactly at sort of the functional neuroimaging of Charles Bonnet. So absolutely, so actually patients who have blindness do get hallucinations. Whether it's the same, it's probably not the same mechanisms, because the fundamental thing that they have is blindness. But yes, that relationship between visual changes and hallucinations, I think is a really interesting one.
Thank you.
Rimona, last question, if you're still up to it.
I could talk about this all day. It's so interesting, and I love the questions. They're like really, really important and interesting.
So Eliran Tal asks, he cites, I think, significantly larger pupil diameters with unequal pupil sizes after light adaptation have been reported in PD.
Okay.
And then he's asking, do you think it is due to the changes in the interhemispheric connections? Yeah, this is his question, I think, but I'm not sure.
It's a great question. I don't know, and I haven't come across that, so I'm going to look that up. I don't know. Fascinating. I mean, there is another effect of Parkinson's, and I alluded to outside the brain, and that's the autonomic nervous system. So there are quite widespread autonomic nervous changes. So if we were thinking about pupil changes, I would probably think outside the brain for differences. But whether, I don't know. There are also reported locus coeruleus differences as well. So maybe both, maybe it's a heterogeneous effect. If there's one thing I've seen with Parkinson's, is it's a very heterogeneous disease. So there could be more than one cause.
Okay. Again, I'm inviting everybody to unmute yourself, and really thank you for a wonderful talk. Very exciting. And have a lovely 2020. Next week, there was supposed to be a conference in Israel, and so there wasn't any talk scheduled, but now due to the COVID situation, it's been canceled, so we may be able to schedule a talk, but we'll notify you about that. And in 2 weeks, we've got Alex Bowers, who will discuss issues related to driving hazards, but I will send specific details about that. So thank you again, Rimona, for a wonderful talk.
Thank you so much. Lovely to see you, and thanks for
to see you too.
people engaging. Thank you.
Thank you.
Thank you.
Bye.